Okay, so for today's accessory video, I'm going to talk to you. We're going to try to do kind of a demonstration of those holiday junction structures that we saw coming up during DNA recombination. This is one of those things where sometimes trying to hold the structure in your hand can kind of help you get a better handle on what's going on instead of just drawing it this way. So let's go ahead and do this little demo. Okay, and so what I have here is just like in the previous video on DNA topology, where we could use some yo-yo string to make our point. Here again I have some string, and this time I've colored each of the two strands of the string with a different color, and I've got that here for two strands that we're going to imagine forming a holiday junction with. So these are double-stranded DNAs, and we're going to form a holiday junction between them. And so one way we can start to think about this is to remember that in order to form these recombination structures, the idea here is that this blue and this green strand are base paired together. And so in principle, we can imagine a little bubble here that's open with the single strands. And we can do the same thing for the other. So presumably, these two pieces of DNA are the same sequence. And so if we open them up and produce the single-stranded version of each one, then in principle, you could have base pairing interactions occurring between one strand of that and the other. So now the purple strand is base paired with the orange strand. And similarly, the green strand could now perform base pairing interactions with the purple strand, right? And so there's nothing preventing us from forming these types of structures that you see here, right? And so if we were to draw what we see here by pulling these strands apart, we would see that we have a blue strand here, a green strand here that now is going to cross over like this, and we've got a yellow strand down here, and we've got the orange strand here like this, and the purple strand, of course, continues onward like so, right? So with these strings here, you can see that this structure that we've drawn consists of two crossovers. Like we've seen previously. And so let's simplify this down a little bit here to one of these crossover structures so that we can look at it in more detail. So if we wanted to inspect a single crossover type reaction, let's do this again, then what we're going to say here is we have our two strands here, and we're going to unwind one of them, the other one, and we will, I guess orange was on the top previously, and we will unwind the green one here as well. So we've got our purple strand, our yellow strand, and we know our green strand is now going to base pair with the yellow strand, and the orange strand is going to base pair with the purple strand, right? And so by kind of zooming in on one of these structures here, we can see something that's starting to look very much like one of these holiday junction type structures that we've talked about in the class. That is, we've got the purple strand, we've got the green strand, we've got the orange strand, and we've got the yellow strand. So now, just with our strings here, we are looking at a single crossover. Okay? And why am I showing us one crossover? Well, one of the nice things that we can do with this configuration here is look at the different ways that the Holiday junction structure could be resolved in terms of the actual strands here. And so let's take a closer look at that. So one way to make it a little easier to see how these different ways can resolve, is to rotate the structure around a little bit so it's easier to visualize what's happening. So I'm not cutting or breaking anything. I'm just rearranging the way that the pairing interactions are to make it a little easier to see what's going on here. So remember, I think if we look at this structure now, all I've done is I've rearranged some of the strands. Maybe we'll twist them up here a little bit to make it easier to see what's going on here. But whereas originally the green and blue were continuously annealed to one another, now the green strand comes off and it starts to form base pairing interactions with the yellow strand here. And whereas the yellow and orange strands had once been continuously base paired with one another, now the orange strand comes off and it makes base pairing interactions with the purple strand. And so if you look at the diagram here, you'll see that simply by rotating this from this version where it looks like a crossover to this version, it's kind of an easier way of, in my opinion, picturing the different ways the different strands are going. And this is also a very convenient way to look at the structure for thinking about the resolution, and you'll see why in a second. And so when we're looking at it like this, and we're thinking about the ways that we can resolve this, we can either imagine slicing here and connecting these fragments together, or we can imagine slicing here and connecting these fragments together. And it should be straightforward enough for us to take a look at the different products that we form. So let's see if we can do that. So I'm going to cut this way first. So I've sort of mutilated my DNA, but now you can see we've made the two cuts here. And if we were to reseal these like this, that would be one way in which we could do that. And if we were to draw out the products of that, we would see that we got yellow and I guess we have to rotate this around. So we had yellow, orange, and now the green strand was crossed over here to our yellow strand, like so. And on this side, it would be pretty much the same purple on top and green and orange, right? So that would be what happens if we were to cut in that configuration. So let me see if I can set it up so that we can imagine looking at the other configuration here. So in that case, just have to imagine that these have been a little ligase action right there. Okay, so we've reconnected those strands together, and let's say if instead we're going to cut this way. across the Holliday junction. Okay, so in this case, I've now cut across the junction this way, and I'm going to seal the products instead in this configuration here. Okay, so by recombining them this way, we can see actually that the distribution on the strands is different, right? And if we were to draw that out, so if we just look at the colors that we get here and we draw them out, we'd see now that we have one that goes purple, and then yellow, and then is green entirely on one side here. And then similarly on the other, we have one that is orange and has the purple and yellow strands like that. And you know, I'm not expecting you guys to know the different outcomes that you get from drawing these, but it is a nice way of seeing what this Holliday junction structure is and the different ways that you can resolve it that lead to different outcomes. And if you were to go on and take, say, a genetics class or like a molecular genetics course, the types of crossover outcomes that you get end up being important for thinking about you know, chromosome maps and genetic maps and things like that, and what types of inheritance patterns you can see depending on how close genes are to one another on the chromosomes. And so yeah, if you want to give it a try, I think it's a good way, I think it's kind of a fun way of looking at these Holliday junction structures. And I do think it, at least if you saw me fumbling around with the strands trying to lay them out and cut them and things like that, it definitely makes me appreciate and respect even more the machinery inside the cell that can perform these same, that performs these processes on DNA.